Did you know that your body has more bacterial cells than human cells? Actually, if you think about it, that means we're more bacteria than we are human. So where are all these bacteria? And why don't we notice them?
Well, they are mainly located at barrier surfaces on your body, like your skin, your lungs, and of course your gut. Normally, they exist in a balance with your own cells, but when this balance breaks down, we become sick.
But not all bacteria are bad. We have many species of good bacteria that live in synchrony with our system in our gut. And these guys help us digest food, they produce extra vitamins, and they also actually directly protect us from bad bacteria by living in the same space that the bad bacteria would live in.
Our exhibit, Gut Reactions, focuses on gut bacteria, how they interact with the immune cells in our bodies, and the changes that occur when disease strikes.
Inflammatory bowel disease, or IBD for short, is an increasing problem across the globe. It comes in one of two varieties, Crohn's disease or ulcerative colitis. Crohn's disease tends to occur anywhere in the gastrointestinal tract from esophagus to rectum, and it causes patchy inflammation that can extend deep into the tissue. Ulcerative colitis causes ulceration, but it's restricted mainly to the colon.
So how does this inflammation start? Well, it's a combination of factors including your genes, the environment, all those bacteria, and your own immune cells. Actually, it's quite amazing that we aren't sick all of the time because there's only a single layer of epithelial cells lining your gut that separates all the microbes and food from some very reactive immune cells. These immune cells protect us from anything foreign, including all of the bad bacteria that can cause disease.
The reality is that occasionally bacteria can breach this barrier, and they probably have in both you and me. However, we don't have inflammatory bowel disease. How is this possible? The answer lies in a special subset of immune cells called regulatory T cells. These cells police the immune system and prevent responses to invaders from getting out of hand.
In some very rare cases, unfortunate people have mutations in a gene called FOXP3. This gene is necessary for regulatory T cells to develop in our bodies. In those patients, they have very aggressive inflammatory conditions that are often fatal.
So what weapons do these regulatory T cells, or Tregs, use to police the immune system? One of their best strategies is to secrete a molecule called interleukin 10, a kind of tranquilizer that suppresses the activity of immune cells. These Tregs are one of the key reasons why we can survive in a generally healthy state, in harmony with all the bugs inside us. In Professor Fiona Powrie's lab at the University of Oxford, we are exploring immune regulation and how it breaks down in IBD and cancer.
Our lab has previously shown the importance of Tregs in controlling IBD using a wide variety of experimental models as well as observations in human patients. Currently, our lab is still focusing on the balance between the immune system and bacteria. One of the ways we examine this is by isolating cells from tissues from IBD patients and cancer patients, and we look at those cells in the lab. We grow them in petri dishes, we try to identify what kinds of proteins they make, how they interact with bacteria, and what kinds of genes they express. And we compare all those to cells from normal patients. And from this information, we can start to glean insights into how patients have developed IBD and how we might better treat it.
We are really lucky scientists because our lab is part of a translational gastroenterology unit in the Nuffield Department of Medicine. This means that we get to work in a comprehensive team that includes nurses, patients, and medical doctors.
That means that we have a lot of direct access to patients in the same facility. So because of that, we can actually obtain a lot of direct tissue specimens and cell specimens from those patients to do really cutting-edge research on primary patient tissue. On the other hand, we can also apply the concepts that we generate in the lab directly to experimental therapies to try to better treat IBD patients.
The relationship between our body's defences and our microbial communities is a fascinating system that's central to our health. Why not join us at the Royal Society exhibition this summer to learn more? We'd love to see you there.